Hello everyone. Today we are going to discuss vibrissae and marine and terrestrial mammals. So first off, what is a vibrissae? Vibrissae are modified hairs. They are semi-rigid and they act as a sensory organ. Vibrissae are a part of the integument or the tough outer cover of a living thing. In animals, the integument includes the skin, hair, nails, and exocrine glands. Vibrissae are a tactile sensory organ, so they aid the animal in the sense of touch. The vibrissae are capable of sensing object distance from the face, orientation, and surface textures of objects. This is possible due to the mechanoreceptors connected to the base of the vibrissae in the skin. This figure here shows the general anatomy of vibrissae. There are extrinsic and intrinsic muscles that are used to move them. Extrinsic muscles, which is this one back here, move all of the vibrissae as a whole. Intrinsic muscles move each one individually. So intrinsic move a single vibrissae, extrinsic move all of the vibrissae. So how do marine mammals use their vibrissae? The two most common uses of vibrissae in marine mammals include using them to gain tactile information about objects in the water and using them to help fan food into the mouth. We will discuss examples of these in detail in the next two slides. For now, here's a photo of a manatee and a seal, and it shows their vibrissae. So the manatee here, you can see they have short, stumpy vibrissae all over their mouth and all over their lips. And these are called bristles. The seal over here has longer ones that look more like the whiskers of a cat. All right, so we're going to start with harbor seals. So for this research, researchers used one male and one female seal. The seals were placed in a pool one at a time, blindfolded with eye caps, and asked to choose the larger of two discs presented to them. Researchers used a set of 22 discs that ranged from 1.12 centimeters at the smallest to 8.74 centimeters at the largest for diameters. The discs were loaded onto a contraption that would fall when the seal pushed on it, which allowed researchers to see which disc the seal had chosen. Researchers tested the threshold of the vibrissae by having the seals compare discs in sizes that got increasingly close together until the seals could no longer tell which disc was larger. The seals were rewarded for choosing the larger size disc with dried fish. Here you can see the graphs made from the data points obtained during testing. So for graph A, this was their smaller size threshold, so they compared disc sizes to the smallest disc. So there's like one constant disc in each trial. This constant was the smallest disc. And on this, you can see that as the diameters got closer and closer together, they did have a little bit more trouble around the 1 centimeter mark, and it went down from there. Trial B here was the medium size threshold. So they kept the medium sized disc as the constant and compared other sizes to it. And this one, you can see they started having trouble around the 2 centimeter mark, but they really struggled around 1. Up here, graph C was the larger disc threshold. So they used the largest disc as the control, and then they compared it to the smaller size discs. And this one, you can see the female had a little bit of trouble the whole time on this one, but overall, they really both struggled at around 1 centimeter difference. So using the data points gathered for these tables, the researchers also calculated a Weber fraction. And a Weber fraction helps tell how accurately a sense can sense something. So to put this in perspective, a human hand has a Weber fraction of 0.03. And the lower that fraction is, the better accuracy that sense has. And a monkey's paw has about 0.1 Weber fraction. So if you look here, the lowest one they found, the lowest Weber fraction was 0.08, which shows that the whiskers of a harbor seal, the vibrissae of a harbor seal, are not quite as accurate as a human hand, but they are more accurate than a monkey's paw at determining the size of something. So next we have manatees, and as we talked about before, manatee vibrissae are short and they're thicker than those found in terrestrial mammals and thicker than those found in the pinnipeds, like we talked about in the last slide. They use these vibrissae to move food into their mouth and to move objects in the environment. So the upper and lower lips of the manatee are covered in bristles, and they move in a way that is described like fanning, similar to how a caterpillar would eat a leaf. The lips of the manatee are bilobed, so you can see up here how it's kind of wide like this. The lobe splits in the middle, so there's one lobe, two lobe, which allows them to move their lips sideways as well as up and down, which can be used to clamp food and clamp objects in the water. So researchers presented manatees with a plexiglass wall with vegetation or 3D shapes stuck to it. So vegetation was stuck in little holes so they could pull it out. The 3D shapes were like screwed on so they couldn't take them off. And there was a camera behind the plexiglass that recorded the lip movements against the glass. So researchers were then able to replay that footage and analyze the movements. It was found that during feeding, the top lip raises and turns outward, which you can see here with these arrows, which exposes the U2 bristles, which are these ones here and here. And these bristles sweep inwards towards the midline of the body, while the lower jaw opens, which you can see here. These are sweeping inward. This is lowering. So then the lower lip curls to expose the L1 bristles, which are on the lower part here. The U2 bristles sweep food to the mouth with help of the L1 bristle, which you can kind of see here. And then the lower jaw closes, which is this arrow here. And the whole process starts over. So the whole process, this whole thing here, from start to finish and over again, takes about 610 milliseconds. Researchers also found that the left and right U2 bristle pads can move independently when a manatee is interacting with objects. So they don't have to move in the same motion. It was also found that the U2 pads could be used simultaneously in a clamping motion. So they were able to like pull things that were stuck to the plexiglass. If a manatee consumed a food item that was deemed undesirable, it was able to use the bristle pads to remove the food item from its oral cavity. So manatees have this prehensile ability in their vibrissae that pinnipeds do not, which is the ability to use these bristles to move things in the environment instead of just exploring the environment like seal vibrissae are used for. And the evolution of bristles in manatees is expected to be linked to the evolution of other feeding features, such as their supernumerary teeth. And the prehensile behavior of the bristles likely formed as an adaptation to allow for better efficiency in grazing on aquatic vegetation. All right, moving on. How do terrestrial mammals use their vibrissae? So they use their vibrissae to supplement the input of their other senses. When an object is too close to see very well, they can use their vibrissae to kind of like face touch the object and get an idea of what it is. Not all terrestrial mammals have vibrissae or have as good a control of their vibrissae due to lack of intrinsic musculature. So here you can see a cat. I think that's what a lot of us would think of whenever we think of whiskers. And then here is a primate. Most primates do have vibrissae. Humans are just one of the two that do not. The other one would be orangutans. So researchers looked at the presence of intrinsic musculature compared to the external organization of vibrissae in 19 primate species and 7 mammalian species that were not primates. Animals were listed as having intrinsic muscles if striated muscles went into the vibrissal capsule. So like we saw on the first image, the bands going into the capsule, the intrinsic muscles. And they were listed as having organized vibrissae if the vibrissae formed a clear row on the face and they weren't just randomly scattered. Researchers used Pagel's discrete method to determine an evolutionary correlation between the musculature and organization of vibrissae as well as the activity pattern of the animal. So they ran 4 phylogenetic analysis, 1 for intrinsic musculature and vibrissae patterned together, then 1 for each of the 3 variables listed earlier, which was in the independent model. The next model looked at all the traits together, which was the dependent model, and the last model compared the independent and dependent models. So strepsirrhines were found to almost all have intrinsic musculature and well-organized vibrissae. So that's one section of the primates. The next section, the nocturnal tarsiers were found to have intrinsic musculature while the others did not. Anthropoids did not have intrinsic musculature. The models determined that vibrissae musculature and organization have correlated evolution. The dependent model did not find any significance, though. However, under closer examination of the model, researchers determined that musculature is dependent on organization into rows. So it is likely that primates lost intrinsic musculature first, then distinct organizational patterns second. Which leads us to our final topic for today, which is the evolution of vibrissae. It is likely that distinct organizational patterns of vibrissae evolved before intrinsic musculature. There was no significance between nocturnal arboreal mammals and diurnal terrestrial mammals, showing that vibrissae organization musculature did not evolve in response to activity and lifestyle. The species with intrinsic musculature were smaller bodied, lived in social groups, and were nocturnal. And the results suggest that intrinsic musculature and highly organized vibrissae were found in ancestral primates, but were lost when vision ability increased due to less need for face touch. And here you can see this lovely figure that was in the primate paper that can show you kind of how they expected evolutionary losses to occur and how they expected gains to occur. So if you look at this one here, it says Q12 and Q21. So from intrinsic musculature and highly organized, there was likely a loss of organized vibrissae in the presence of intrinsic musculature, which is how we got to intrinsic muscle with lower organization. Whereas going back, there was a gain of organized vibrissae in the presence of intrinsic musculature. So I thought this was just a really cool thing to look at and kind of helped explain the connection between the evolution of the intrinsic muscle and the organization pattern more. So if you have any questions, please add them to the discussion board below. I look forward to hearing from all of you. Thank you for watching my presentation.